prediction and improvement of better pearl millet genotypes. So, myself, Dr. Harshraj Shinde, I'm postdoctoral scholar in University of Kentucky. So currently I'm working on genomics and epigenetics and genetics group in under Department of Horticulture here. So during my PhD, I work on pearl millet. So here I will speak mainly about the salinity trait and how genomics we applied to study the salinity stress in pearl millet. So this is the outline of my talk. So in introduction, I will give about introduction about salinity stress. RNA is related to salinity stress. And finally, I will speak about one of the transcription factor gene, that is NAC21. This gene we discovered in our first study, that is transcriptome analysis, and then we carried out the functional analysis of this gene in Arabidopsis, and this gene proved to be a salinity stress tolerant gene. And then finally, I will conclude my talk. So salinity, it is mainly caused by a combination of sodium, magnesium, calcium, and other ions in the soil. So it has estimated losses in agriculture. Yield losses in agriculture due to salinity are 16%. Globally, a lot of land is affected by salinity stress. 20% of irrigated soil is affected by this stress. So in this picture also, you can see the how salinity stress has caused the soil problem and in rice. So if you go into the global picture, a lot of soil at global level has been affected by salinity stress. Right from here, Middle East area, Middle East Asia to Southeast Asia, most of the soil here is highly affected by salinity stress. Speaking about India, still the percentage is low as compared to the other Asian part. In case of Australia, a lot of soil is affected by moderate amount of saline soil. So salinity is the global problem. Then I will speak about the crop which which we studied here, which is pearl millet. The botanical name is Pennisetum glycom. In Marathi, we call it as a bajri, and in Hindi, we call it bajra. So it is staple food crop for more than 90 million people in Africa and Southeast Asia. In India, also a lot of farmers, they are dependent on pearl millet for their food and survival, especially in Rajasthan, some dry areas of Gujarat, even in Maharashtra. So it's kind of important crop. If you go the study wise, the number one is rice, number two is wheat, number three is maize, the number four is barley, and then it comes the pearl millet. So it is one of the staple food crop. It is originated in West Sahara zone, which is very hot. It is diploid. It has the diploid chromosomes with 2n is equal to 14. And in 2017, Rajiv Varshney group of ICRISAT, they sequenced the genome of pearl millet and they estimated the 38,000 genes in pearl millet. And it has very high photosynthesis capacity as compared to other crops. So the main feature of this crop is abiotic stress tolerance. It is drought tolerant crop, so it can survive at 250 mm of rainfall. It is high temperature resistant or heat resistant crop, so it can flower at 42 degrees Celsius. It is a salinity tolerant crop also, so it can survive up to 250 millimolar of NaCl of 250 millimolar of NaCl. So my main topic is on salinity stress here. And if you go into the nutritional value, it has very high or very good nutritional profile. And if you compare it with rice, it has very high protein, almost 1.6 times more proteins as compared to rice. High amount of fibers. It's good for, that's why it's good for digestion. High amount of minerals and almost 30 times more higher amount of iron. So it's good for pregnant and lactating mothers. It has 4 times higher, 3 to 4 times higher calcium content. So again, it's good for infants or lactating mothers. So if you go into the nutrition value, it is highly nutritious and good nutrition profile crop. But despite of having all these characters, pearl millet is one of the least studied crop. If you go into the number of research publications, so as compared to rice, wheat, maize, or barley, or other cereals, it is one of the least studied crop. Since the genome sequencing in 2017 by ICRISAT Rajiv Varshney group, its a research in pearl millet has been advanced. Many scientists and researchers has been attracted to this crop. So aim of our study was to study the physiological, molecular, and biochemical responses of pearl millet to salinity stress. So in case of molecular responses, we perform the transcriptome analysis. Very few biochemical responses we studied. Physiological responses of different pearl millet genotypes were also studied. The genes which we got in transcriptome analysis, that is in RNA sequencing, which studied one gene. Another students in Japan, they are studying now another genes. Then functional analysis of such stress tolerant genes. So I will start with my study one. First one is the comparative de novo transcriptomic profiling of the salinity stress responsiveness in contrasting pearl millet lines. Here we perform the RNA sequencing of different pearl millet lines. So material and methods or seed materials of pearl millet. So initially in 2016, we got the seeds of pearl millet genotypes from ICRISAT. So especially pearl millet breeding station, Dr. Shashi Kumar Gupta, they provided us the pearl millet seeds for this study. So they provided 8 lines, 4 were salinity tolerant lines and 4 were salinity susceptible lines. Then we conducted some of the trials in greenhouse for salinity stress tolerance and susceptibility to have the perfect lines for our future RNA sequencing experiment or transcriptome studies. So we, based on our initial screening in greenhouse, we selected ICMB 01222 as a baseline or perfect salinity tolerant line. And at the same time, we selected the ICMB 081 as a perfect susceptible line to study the contrasting nature or contrasting phenotype during salinity stress. So after selecting these 2 lines from our initial trials, so we again perform the physiological analysis of these 2 lines to compare them. So we grow both these lines in same pot. So here you can see that this left panel is under control situation. So you can see under control situation, both these lines has almost no effect. They are growing healthy, but after like 16 days when we applied stress, salinity stress, By giving them the 250 millimolar of NaCl through irrigation channel, you can see the clear difference. You can see the salinity tolerant line is still healthy, almost no effect on their leaf, no effect on their height, little effect on their height. But on other hand, if you see in the salinity susceptible line, that is ICMB081, the leaf started drying, lots of symptoms like deep burning, loss of chloroform, yellowing we observe. So we, based on this analysis, we think that both are the perfect tolerant and susceptible lines for our future studies. Then we perform some of the biochemical responses of physiological responses of pearl millet to salinity stress. So we measured the relative water content, and you can see that during stress, tolerant line had higher relative water content as compared to susceptible line during stress situation. Although the relative water content was decreased in tolerant line, but it was decrease was very high in susceptible line. At the same time, we calculated the chlorophyll content by SPAD meters, and we found that chlorophyll content was very less in case of susceptible line during stress. And in previous picture also, you could have seen that they almost showed the yellowing, that is almost there was the loss of chlorophyll. But on the other hand, tolerant line had still higher chlorophyll content than susceptible line. And this was our key finding about the sugar. During stress situation, the sugar was increased in both genotypes, in tolerant and susceptible. I think we think that sugar is the key here. Tolerant line showed almost 1.6-fold higher amount of sugar during stress. In case of susceptible line also, sugar increase, but the amount increase here was very less as compared to the tolerant line. And then same again, during stress, higher the sodium accumulation in leaf, the less susceptibility of that line. So you can see the susceptible line has accumulated high amount of sodium in leaf. And we also observed sodium injury symptoms, and so some of the sodium injury symptoms like leaf tip drying, leaf yellowing, and stunting were observed only in susceptible line. Then based on some of the physiological analysis and biochemical analysis like relative water content, chlorophyll content, sugar content, and some of our phenotypic observation, we selected these two lines and we performed the RNA sequencing of both these lines, and we maintained the 3 replications. We selected the leaf samples from both control and salinity stress plants, and we selected these and we sequenced them using Illumina HiSeq platform. After sequencing, we got raw reads, and then we filtered the raw reads into clean reads. We checked and we converted them into the contigs to find out the genes, to find out the genes. And after finding the genes, we performed the differential gene expression analysis. Of course, here our motto was to find out the genes related to salinity stress in both lines. So we performed the differential gene expression in R using edge test, and around 2,900 genes were upregulated. The same amount of genes were downregulated in tolerant line, that is ICM0122. But in case of susceptible line, less number of genes were upregulated and higher number of genes were downregulated. So we think that this is the key difference. The more the upregulation of genes, the more or better response of these genotypes. So here you can see that there was less amount of upregulation in susceptible lines. Then we draw some of the common genes between these two genotypes. So these are the commonly upregulated genes and common downregulated genes between these two genotypes. So after finding so many genes in both genotypes, we went for annotation of genes to find out the specific gene families. So we started with NAC transcription factors. Sorry, we started with transcription factor families identification. A lot of families of transcription factors we identified, but here we have shown only top 10-15 families here. So most uh, the the family which we observed most in the involved in sanitary trace of palmillet was Jingfinger. This was the uh, this family has the most number in palmillet during sanitary trace in both tolerant and tolerant and susceptible. Other than that, we found some of the other families also, that is APT transcription factors, MIB transcription factor, NAC transcription factor, Werke transcription factor, and W8 transcription factor. And there were lots of other transcription factor families also we observed. But the key difference between tolerant and susceptible lines we observed was squamous of promoter binding transcription factor, because this transcription factor family shows expression only in tolerant line ICMB0122. Here we have also highlighted this family because this family was only observed in ICMB0122. So future studies are needed to study this family in detail. After finding the transcription factors, we went for ion transporters identifications, because during salinity stress, ion transporters plays key role in ion detoxification. So we found around 6 families of transcription factors. First one is ABC transcription factors, which ATP binding acids and high efficiency potassium transporters, sodium hydrogen exchange, and other transporters also. So here also, if you see, this transcription factor shows very high upregulations or very high expression in tolerant line as compared to susceptible lines. So here again, I would like to emphasize that more the expression of transcription factor, the better tolerance of that genotypes. So here you can see almost all families showed very high enrichment in tolerant line ICMB0122. Then we perform the cake pathway analysis to find out the exact metabolic pathways involved in salinity stress on pearl millet. We found 300 pathways, but it was difficult to show all 300 pathways here in our analysis. So here we have shown only 20 best pathways or 20 most significant pathways. So different pathways, here pathways showed by different colors for susceptible, tolerant lines, the down-regulated and up-regulated pathway. This panel is down-regulated pathways and this panel is up-regulated pathway. So among pathway, the key difference was the glycolysis and ribosomes, then phenylpropanide and ubiquitin-mediated proteolysis pathways, because these pathways shows very high. For example, glycolysis and gluconeogenesis, this pathway shows very high amount, very high down regulation in susceptible genotypes, but this pathway were almost showed very less down regulation in tolerant genotypes. So this pathway were also a key pathway. And on other hand, the phenylpropanol biosynthesis and ubiquitin mediated proteolysis, this pathway shows very significant upregulation in tolerant genotype as compared to susceptible genotype. So we think that this pathway also holds key for stress tolerance and susceptibility during salinity stress. So key finding from our this study was the tolerant genotype ICMB0122 showed higher growth rate and accumulated high amount of sugar in leaves under salinity stress. Squamous promoter binding transcription factors were only found in salinity tolerant line. Ubiquitin mediated proteolysis and phenylpropanol biosynthesis were upregulated in the tolerant line. And we think that tolerant line can be used as a donor lines for the donor, as use can be used as a donor of useful genes to further improve the salinity tolerance in pulp millet. So our next study is the small RNA sequencing to find out the microRNAs related to salinity stress. So here, based on our previous studies, we selected only tolerant genotype. We got some of the raw reads and clean reads. After sequencing, then we went for differential expression of microRNAs. So in this study, we found 30 upregulated and 51 downregulated microRNAs. Among these microRNAs, we identified the microRNA 159 was the most significant family in case of silent stress. Then we went for the target gene identification for these microRNAs, because microRNA depends on their target genes for their functioning. We identified different families of target genes. Among them, squamous promoter binding and MIP transcription factors were the most enriched target gene families. Then we also went to find out the pathways which microRNAs regulates during salinity stress, and we found that during salinity stress in pulp millet, microRNAs and their target genes regulate the oxygen signaling pathway. Here you can see that this upregulated and downregulated microRNAs regulates many oxygen oxygen signaling pathways genes. And these genes finally goes for salinity stress response during salinity stress. Then we also validated the expression of these microRNAs by qRT-PCR. Almost by in our qRT-PCR results, microRNAs showed the almost similar expression pattern like RNA sequencing. Then we also performed the qRT-PCR analysis of microRNAs and their target genes at the same time. These are the microRNAs and these are their target genes. And in our analysis, we found that both these microRNAs and their target genes shows contrasting expression patterns which we expected. So key findings of our studies are 81 microRNAs we discovered in pulp millet which are salinity stress responsive, and we identified 448 target genes for these microRNAs. Around 25% of them are transcription factors, and these microRNAs regulates oxygen responsive pathway during salinity stress. And we think that some microRNAs plays role in salinity stress tolerance mechanism of pulp millet as like other crops like Arabidopsis and rice. Then our final study, we studied the NAC transcription factor of pulp millet, that is PGNAC21, and we found that this NAC transcription factor enhances the salinity stress tolerance in Arabidopsis. So this NAC21 we found in our RNA sequencing analysis. So NAC are important family genes, and here we perform the functional studies of this gene, NAC21 gene. We perform the gene expression analysis of NAC21 during salinity stress, and you can see as time of salinity stress goes, after 6 hours of salinity stress, it was upregulated. After 12 hours, it was still high upregulated, but during 18 hours of salinity stress, it shows almost 24 to 5-fold change. So based on our RT-PCR analysis, we think that this gene is best candidate gene for future studies. And we also found that this gene's expression is also upregulated by ABA treatment. ABA is the abscisic acid and stress hormone. So that's why we selected the expression, we selected ABA for our analysis. Then we constructed the transgenic lines of Arabidopsis, which shows the transgenic lines of Arabidopsis, which overexpresses the pulp millet NAC21. And then we perform the salinity stress tolerance assay of this transgenic line. Here This is the control condition. You can see all plants are growing well. Col 0 is the wild type. This is the transgenic plant line 2, transgenic line 6, and transgenic line 3. So here in this picture, you can see as compared to wild type, the transgenic lines are showing high amount of germination. Then their fresh weight is very high. So based on our these studies, we think that NAC21 is the best candidate genes to study the salinity stress in pulp millet. So this is one of the one of the key candidate gene. Then for same NAC21 overexpression lines, we perform the root assay. So you can see during the salinity stress, overexpression line showed the very high amount of root elongation as compared to the wild type lines. So we think that this gene is also responsible for root elongation during salinity stress. Then we find out some of the target genes of NAC21 in Arabidopsis. Most of these genes are stress responsive genes. And then we propose the model how NAC21 works in pulp millet. So key findings of our studies are expression of NAC21 is upregulated by stress, salinity stress, and ABA. MIB transcription factor bind to the promoter of PGNAC21. These results we have not shown here. And I have shown the result of transgenic lines. So transgenic lines of Arabidopsis, those who overexpress the pulp millet NAC21 has exhibited the stronger tolerance to salinity stress than the wild types. PGNAC21 overexpression Arabidopsis plants also has shown the enhanced expression of stress responsive genes. So finally, conclusion from our all three studies. Understanding the effect of salinity stress on pulp millet and their mechanism of salinity stress tolerance are vital for breeding new salinity stress tolerant cultivars. Based on our RNA sequencing study, we found that the total soluble sugar content was higher in salinity stress tolerant pulp millet lines, and sugar metabolism related pathways were highly upregulated in the tolerant line. So we think that the sugar is the key. Higher the sugar, higher the salinity stress tolerance in pulp millet. MicroRNAs also mediate the stress tolerance in stress response in pulp millet. So, and we found that they regulate the stress response in pulp millet through oxygen responsive pathway. NAC21, one of the genes which we studied, is also the key stress responsive gene and is the positive regulator of salinity stress tolerance. So finally, I would like to acknowledge ICRISAT, International Crop Research Institute for Semi-Arid Tropics, India, for providing the pulp millet genotypes. And also special thanks to Dr. Shashi Kumar Gupta for their guidance for pulp millet different assays. We would like to acknowledge Nodai Genome Research Center of Japan. They funded us for RNA sequencing analysis. Finally, I would like to thank the University of Tokyo, Japan, where I did my PhD and where all this work I carried out. And also thanks to University of Tokyo for providing Todai special scholarship to me. So based on our all three studies, we have published 3 papers. So these are the titles of all these 3 papers. First one is for RNA sequencing, second one is the microRNAs, that is small RNA sequencing, and third one is NAC transcription in NAC21 studies in pulp millet. So among these papers, microRNA papers has been published recently last month. So if you have comments, questions or suggestions for our future directions, these are my email ID. You can send me emails for any kind of suggestion, comments or questions. Thank you for attention. I hope you enjoy my talk. Thank you.
And so working as a assistant professor cum research scientist in IMIM Institute of Agriculture and Technology. Okay, the first and foremost, thanks to my team members to conducting this sustainable agriculture for future and present mankind. So, what are the innovative technologies we need to adopt and to make good, healthier life? Okay, so in this section, first I am giving a short introduction about the topic. So, the sustainable agriculture is foremost important how we need to run sustainably our life through growing our plants. Also, the current and future need for agri and horti technology. How the sustainable agriculture to linking between the production productivity of these crops. Okay. Also, we need to learn what are the farming tools should learn to make the sustainable agriculture. Okay. So this is my email ID and my contact. Okay. So, first, what are the innovative technologies to make healthier life as well as the sustainable life and also the sustainable agriculture? So, I'm listed, there was the 9 important technologies and 9 mantras for making sustainable agriculture for the present and future generation. Okay, the first one is the production of nutrient dense fruits and vegetables. The first mantra is nutrient dense fruits and vegetables. Next one is high density planting. Okay, then permanent agriculture, that is called permaculture. Then production of microgreens and sprouts. Then floating agriculture, barter technology, application of mobile app for exchanging of food and growing technology. Then need for community garden. Then the foremost important post-harvest last food grains and fruits and vegetables. How to make fruit jelly, how to make homemade jam, homemade squash. Yes, we should know the technologies. The foremost important 3R technologies, that is reduce, reuse, and recycle. These are the 9 mantras to make a sustainable life as well as sustainable agriculture as well as to safeguard our earth from the natural disaster, this pandemic situation. So we need to think over the 9 mantras. I'm going to describe the individual technologies. Okay, the first technology is the production of nutrient dense fruits and vegetables. The name nutrient dense. Why we want to eat nutrient dense fruits and vegetables? Yes, we are getting day to day life all the fruits and vegetables from the market. How many of you know the nutrition profile of individual fruits and vegetables? How much of where it was grown, which soil it is grown, doesn't know. If you buy the biscuit packets, if you buy the other goods material in your supermarket, everything is there. The nutrition profile, this much carbohydrate, this much protein, this much calories, this much vitamins, everything they are listing the profiles. What about our fruits and vegetables? We never think about where it is grown, what is the nutrition profiling, what we are eating. We are never bothering that things. How will be a healthier life? Yeah, in our globe having diversified soil profile, diversified fruits and vegetables. So why we want to think about the nutrition dense fruits and vegetables? That is the foremost important for soil health management and plant health management, our health also. The triangle is there. So if you take an example, apple, they planted in 1950s in a Himalaya region. They take the 1950s, after 2 years they make the fruits. They take the fruits, they studied about the nutrition profiling, what are the components, what are the vitamins are there. Then they made the nutrition profiling after 50 years in the year of 2000. The same tree and same apple, the nutrition profile entirely is changing. Why it's happening? The changes of nutrition profiling. For example, vitamin C, 100 milligram per 100 grams of fruits in the year of 1950. But in the year of 2000, the vitamin C is now 20 percentage. But the apple size is there, apple color is there, the growth and development is there, yield is there, but the nutrition profile is changed. Then we are consuming less nutrition fruits and vegetables. Why? We are not even bothering about the soil health and soil microflora and fauna as well as the rhizosphere beneficial microbes. We are throwing like anything fertilizers and other chemicals to killing the microbes as well as to killing our soil health. If you're killing the soil health, then automatically it should be reflected in your outcome of the soil, fruits and vegetables. We are consuming very nutrition less fruits and vegetables. Then how our life should be extended, then how to fight for this pandemic situation for COVID-19. So everyone to take care of the soil health. Then only now their standing changed, grow by one from their farm by where it was growing. Everybody we need to make geotagging as well as the nutrition profiling. So the peoples will consume to know what are the things we are taking in healthier life. We should know the production of nutrient dense and fruits and vegetables in future as well as current situation. We need to think about this diversified soil condition. We need to think about the production of fruits and vegetables, even food grains should be a nutrient dense concept we need to follow. This is the foremost important for soil health. Soil health is okay for the sustainable ability will be there and a sustainable agriculture we can run it. This is the first foremost mantra for soil health. Next, we want to grow. We want to feed our generation. We want to feed our population. So day by day increasing the population. We are the second place in globally for the population. Now China is leading in the first population place. They are also producing fruits and vegetables. So we need to feed all the common people. The technology will to change. High density planting, that is HTP technology, now they are practiced now implementing banana, pomegranate, mango and guava as well as vegetables. The land use efficiency, agriculture inputs and land use efficiency, and to make the protection of fruits as well as vegetables for making high density planting. Then only we need to succeed. Production and productivity is there, but each and everyone's healthier life means we go for the high density planting methods to produce the vegetables. So if you take in a classical example, we are in the Cavendish group 63 percentage in our Indian scenario. Other bananas also is there, but the national wide as well as globally, the Cavendish playing a major role. Yeah, banana, you know the 56 nutrition profiling there. All kind of amino acids, carbohydrate and proteins, vitamins, everything, iron, minerals in rich in potassium. So each and everyone they consume a daily one banana, the healthier life should be there. So how the innovative technology, we need to interfering the biotechnological tools, how to make the mankind, so that conventionally the farmers they are doing sucker cultivation, also the stereotype culture. So now the innovative technology for making one cell line initiated for 80,000 to 1 lakh plants, we can make it. Why? We are high density planting, we need more planting materials. The planting materials should be disease free as well as pest free, high yielding, faster rate and more yielding uniform harvest, so that we need innovative technologies like the embryogenic cell suspension technology to make and produce bananas, and making the planting materials for most important the situation. So we want to avoid the disease incidents, avoid the pest. We need to go for higher rate of multiplication and healthier plantlets, and also the sustainability. So 100% yield and income as a farmer, we are growing, but yield and income. So we need to take care from cell to harvest, not from the plantlets to the harvest. We need to take care the cell to harvest. From also the stage specifically, we need to apply the cultural practice as well as nutrition, stage specific nutrition, both organic way. So we need to attain the yield. So that is the foremost important. So now we want to sustainability means as well as the production productivity to lead and feed our common people. We need to take care each and every plants from sale to harvest. The protocol and SOPs to follow, we can attain the agriculture production. Also the multi-tie system. Yes, nowadays the land use efficiency is, we need to use the land as well as the multi-site system we return for revenue generation. So we need to cyclically we cultivate our crops. Then the weekly income generation is the foremost important for all. If you weekly income generation for agriculture sector, then only we need to sustainability. Yeah, the intercultural operation, manpower and labor charges. So weekly we need to produce something so that alternate we want to grow multi-tie system, jack, apple, orange and pepper. So all the crops, even vegetables also we need to plant multi-tie system. So we need to get revenue generation for the sustainability. So that is the one of the mantra for multi-tie system we'll need to follow for this, as well as quality of disease material, planting material also important for sustainability. Then we need GAP. What is GAP? Good agriculture practice for each and every crops. Yes, I am planted. It was growing. We are applying too much of phenomenon of fertilizer, pesticide, even water. We need to use the good agriculture practice. So if you take one, for example, classical example, banana. Okay, this is the first planted. Next mother, next daughter, next daughter, next daughter. The same land, same cultivation. For example, this is bunch emerged. Next plant is ready for next 2 month intervals to other bunch, another bunch. For within 2 years, we make the 3 harvests. So similar like all crops also we need to make a good agriculture practice. We can more returns, doubling the income of the farming communities. To make GAP practice is foremost important for all kind of crops, to routine income generation with the less usage of water. So interference of the foliar spraying application, avoiding weeds, also using mulch for soil water conservation strategy, we can produce the fruits and vegetables in proper way with the help of GAP practice. This is also foremost important for current and future agriculture. Next, we need to do small intercultural operations. That should be lead for our import as well as export. Our country GDP will be increased. So this is a classical example. All the crops, for example, pruning and training the time period of, for example, pomegranate in North India. So we need to do the timing of pruning for proper flowering. Then only we'll expected yield should be there for small cultural operations to lead to increase our GDP of a nation as well as production of quality fruits. We can make export quality and the local consumers would be healthily we can given to feed our current population. So that is the foremost important for the good agriculture practice and post-harvest technologies, and take caring the produce is foremost important. Okay, next the mantra, great mantra is permaculture. Jiffy Laden, okay, he is one of my friend from Australia. So what is permanent agriculture? Yes, we talk about agriculture. What is permaculture? Permaculture means perma and agriculture. That is permaculture. So what he driven? So he drive 365 days, make the schedule. Today I am consuming apple. The same day, next month I want to consume the apple. Today I am consuming some green vegetables. Same vegetables to consume the next month. So 360 days he having the scheduling to grow as well as to consume the fruits to extend the life of the human, as well as the main concept for this. See, look over the picture. There was the pond, where was the greenhouse, there was the vegetable cultivation. Here also see the rotationally they are cultivating fish, pond, cattle. So these communities never bothering about the pandemic situation, never bothering about the future, never bothering about the shopping rate. They are all involving to growing the own. They are supplying all the homes. That is sustainability. This is a permanent structure they made. From fish to meat to egg, even vegetables, leaf vegetables, everything They are growing by their own. That is permanent structure. That is permaculture. 365 days we need to grow. How we can make 365 days to grow all kind of vegetables, all kind of things? So he is the man is driven. This method is called permaculture. So cyclically, this is one date, the next routinely they will grow the vegetables, even greens, all kind of plants they are getting. So if you take, the lady was standing here, this is the reproductive stage, flowering stage and fruiting stage. The same crop is now the vegetative stage and juvenile stage. Once it is harvested, next it was ready for the harvest. So this today I am harvesting, the same day onion I am consuming, same next month the same thing. So they cyclically they are cultivating and generating their own food. That is called permaculture. That is now the integrated farming system, same approach, but a systemic way of cultivation of crops and vegetables. This also the pandemic situation will be fighting against. If you happening World War III, what will happen? Never moving of, we are lacking of petrol and diesels in India. So a situation, if you grow like this within 5 cents or 2 cents in the city's life as well as in the outside in a village, the same one village can sustainably grow their vegetables and fruits to run their life. So everything in your plate. Okay, that is my plate concept. My plate should be everything grown by their own home they can feed. This is permaculture model for all kind of trees and fruits and vegetables and even chickens and meat and even for egg production, full milk production, even grains, even greens, everything they are growing their own and taking consuming. This is my plate concept for sustainable life as well as sustainability of agriculture. This should be transformed from our Indian situation to marking the population to feed with the low cost with the high energetically food we can consume. Okay, next, the small technologies, that is microgreen and sprouts. The mankind can able to grow the greens at the home itself as well as the sprouts also. The sprouts and greens have rich in vitamins A, D, E, K as well as minerals and potassium, also all kind of calories. So the pandemic situation as well as a new emerging disease we are getting to fighting for this new emerging disease through food. Okay, so if you consuming the greens and sprouts is the foremost important to make immunity life to fighting. These are the technology they can easily do at home itself. Homemade grown up greens and sprouts. This should be useful for the sustainable life as well as the sustainable agriculture at the home itself. So each and everyone, so radish is there. For example, mustard, if you sprinkle the mustard seed in your compost, so then grown up within 7 days, they are ready for harvesting and to feed. As well as sprouts, within 2 to 3 days, make the sprouts and consuming daily, weekly, twice, 2 sprouts and 2 greens. Automatically our immunity will be increased as well as we are working efficiency also increased. These are the agriculture technology now needed for all the conditions. And also the food exchanging. Yes. So we are using enormous petrol and diesel for transporting our fruits and vegetables one place to another place. This is the technology, the intervention of ICT technology in agriculture. Make the mobile app. So one village, yes, one street I am growing tomato, one street I am taking for brinjal, one street I am taking for carrot, one street I am taking for greens. Yes, my street having this much amount of greens available, this much kilo of brinjal is there, this much kilo of tomato. We can exchange within walkable distance to exchange the material. Each and everyone share it, avoiding usage of petrol and diesel. This should be reflect our conservation of our non-renewable resource for our country, as well as to avoiding the soil, avoiding the air pollution by using of vehicles. So each and every village should take the model of this practice, the permaculture practice, as well as rooftop gardening, to exchanging the fruits and vegetables with a low price and each and everyone sustainably to consuming the vegetables and greens at in a village, at in a villas, at in a home gardens to make this. This is the batter means the forefathers, we are forefathers, they did the practice at home. If I am growing too much of fruits, they while coming them will supply, they will give them. While they having too much of fruits and vegetables, they will exchange. So if you take exchangingly, they are getting energy as well as the knowledge of this. Also, each and everyone know the production technology, then to avoid the petrol usage. Next, the foremost important community garden. Yes, nowadays the township kids, even township, the people doesn't know how it is growing, where it is growing, how the plant appears, looking. Yes, in our one of my friend is in abroad, so in US, weekend they are going for growing vegetables. This is a rental based plots. Yes, rental based plots are available. They can grow everything. Weekend they will go and watch. Remaining days the farm will be taken care by the owners, and the rentals they will come and cook, they will harvest in freshly. So the community garden concept for each and everyone should know the reality of growing of fruits and vegetables. The upcoming, the future generation is the childhoods, is the future of our generation. They should know the real agriculture knowledge, to feed the agriculture knowledge to the young minds. The young minds playing the future Indian agriculture sector as well as the Indian mankind. So this should be practiced in our nation also. If you take village side, no problem, the students will learn. In township, we to make the transforming agriculture to the community garden, to exchanging of knowledge. That is the foremost important for sustainable life and sustainable agriculture. So we need to know the compost, we need to know the soil and which plant, carrot look like this, potato look like this, greens look like this. Everyone sharing their knowledge and growing to eat healthiest food. So this should be transforming from the current scenario of mankind. Okay. Next, everyone should know how to prepare jam, jelly, squash. We want to avoid the post-harvest losses. We are producing too much of potato, too much of tomato, less price. Each and every home should know that fruit processing technologies, even the farmer should know the fruit processing technology, post-harvest technology, storage methods, extending shelf-life methods, should learn and sell their products in off-season, as well as the return should be more. So everyone should know the post-harvest losses and post-harvest processing technology for making pickle, jam and jelly. This is also very, very important for sustainable life and sustainable agriculture. Okay. Next, nowadays everybody having diabetes. Everybody want white, pure rice. While polishing, see the percentage losses of allura and layer. Pure white rice nowadays consuming all the peoples. Diabetics 1, diabetes 2. So how can make the sustainable life go for unboiled, brown rice, high fiber rice, white rice? If you take in the electron microscope structure, the comparing the raw and cooked rice for the unpolished and polished rice, we are losing all kind of fibers and minerals and vitamins also. Only we are consuming too much of carbohydrates. So how to change the people mindset? So only the way to know, yes, each and everyone is unpolished rice having high fiber, as well as use of traditional rice. So traditional rice, we have enormous genomes available. Go for traditional rice cultivation and feed our generation in healthier life. This is I grown my farm, we are consuming daily in our home. This is traditional rice. This is taken by the kings for having more fibers. Okay, if you compare the normal rice and traditional rice, they are having too much of fiber and crude protein and dietary fiber to reduce less carbohydrate and less, okay, total amylase also is the starch content also less. This should be increase our longevity as well as for feeding our future generation to practice our childhood age also from traditional rice, unpolished rice also very very important to avoid the endemic pandemic situation of diabetics, also the future big problem, also the current problem. We need to practice in our home, as well as farmers should grow for the traditional rice and traditional plants with the rich in minerals and nutritious food should be grow and also to spread over every place. So we have enormous rice genomes also available. Traditional rice having rich in antioxidants and protein, fiber rich in that. This also we need to promote to cultivate our farmers and feed and the common people. Also, this is the foremost important. So the next, the finally the 3R technology. So each and every one, the compost preparation, alternative energy, solid waste management. So reduce, reuse, recycle. Reduce means if you are going shop buying, if you buying any goods and vegetables or even anything, they are giving carry bag, plastic carry bag. If you each and every home, they are all the home they are dumping like anything the plastic bag. We want to reduce, go for cloth bag while repeatedly buying. Then the same cloth bag we can use reuse. Then it is a recyclable one. So why we want to safeguard our health? Where the health, soil health, we need to take care of the soil health. And also for throwing our homemade waste. Every home and every farm they should know compost preparation, own compost preparation, enriched compost preparation to enrich our soil health as well as to avoid the pollution. Solid waste management, see the big problem, throwing, dumping in line the soil. Soil pollution is the major problem. So every home they having the composting methods to avoid the solid waste management, to avoid the soil pollution. So that the first foremost important the composting technology for sustainable agriculture. Then alternative energies used for solar, for pumping of water, solar for home purpose. We need to go for alternative energy, avoid electrical generation, nuclear generation. So that now the government is implementing for solar and wind energy for agriculture sector as well as this. Next, for traditional plastic and bioplastic, we need to promote the bioplastic. We need to degradable bioplastics. So avoiding the solid waste management strategies, as well as we need to take care of soil health. Then the soil health is okay, the plant health is okay, finally our human health also okay. So finally I am talking about the health condition of the soil reflecting in our agriculture and mankind also. Okay, so sustainable lifestyle to make each and every one. Okay, this is the 3R technology. Yes sir, the finally I'm talking about floating agriculture. What happening nowadays? The climate change. Yes, we are accepting the climate change. Rainfall failure. Yes, rainfall failure. Drought situation happening. How to overcome? All the rains happening at in the ocean, not in the land places. So the floating agriculture is the future and present. We have enormous ocean place. So we need to build a rainwater harvesting unit. So now the composting technology is available. Take the compost and growing seeds also is there. Natural light is there. Enriched compost is there. Good quality of rainwater is there. We need to cultivate, to harvest, then we feed the future population. So the floating agriculture practice they are going to implement very soon due to the scarcity of water and drought, as well as pest and disease. So this is the floating agriculture they are practicing. So I got information from one of my friend in the Japan. They are planning and they did a small experiments. They got the good results. This technology is going to replicate for the sustainable life and sustainable agriculture in this ocean itself. So these are the new technologies we need to replicate in our nation for growth and development. Okay, so finally I am concluding the 9 mantras is foremost important for sustainable life and sustainable agriculture. So intervention of biotechnology, intervention of genomics, as well as intervention of all the technologies for the community people, awareness for growing of good, rich, nutrition food. So we need to consume to we fight for this pandemic situation. So that we need to make future plan. So implementing all the technologies. Okay. So that is the finally I'm concluding. Any doubts or any relevant doubts related to sustainable agriculture, other things, please email me, and also through WhatsApp me. Even anytime you can call and get clarifying from the doubts. Okay. So this is for the current and future need for the science and technologies. Okay. So the nine mantra is there for the production of nutrient dense fruits, high density planting, permaculture, green sprouts and floating agriculture, community garden, 3R technology, post harvest technology should be the innovation should be implemented to the agriculture. Okay, so thanks for the opportunity. Thank you.
Health prospects. This is our groundbreaking team at ICRISAT, headed by Dr. Janila, principal scientist and groundbreaker, and then scientific officers, visiting scientists, postdoc fellows, students, and then researchers. I'm representing here on the behalf of them, so thanks all my teammates for this wonderful contribution. So groundnut, it is also called as one nut, as it is one nut and many stories to tell. So one of the stories is George Washington Carver, whom I admire a lot for his motivational and inventional discovery, in which he has deciphered as many as 300 different products of the groundnut. Here I try to summarize the different uses of the groundnut. So yellow color represents the food uses of the groundnut. It's starting from the curries, the fried groundnut, oil, roasted, butter, salad, and so on. Green color represents the animal uses of the groundnut, that is horn, which is used as a fodder or foliage, then silage, then shell, cake. So all these are the animal fodder uses of the groundnut. Then gray color represents the soil uses of the groundnut. So organic, as it is a very uh, very uh, like bulky crop, it has got much residues which increases the organic carbon content, and then being a leguminous crop, it fixes the nitrogen as much as 80 kg per hectare. Blue color represents the industrial use of the groundnut, starting from the heavy metal lubricant, heavy metal absorbent to lubricant, paint, filler, and solvent industry. Groundnut is very nice blend with oil about 48%, protein about 25%, vitamins and minerals. And that's why it's wonder nut, as it is a blend of good amount of fatty acid, that is healthy fatty acids, and minerals and protein as well. If you look at the overview of groundnut production in India and then world, so India is topmost in the production and then second in the productivity. And apart from India, groundnut is being grown in as many as 118 countries, covering 28.5 million hectares with annual production of about 45.9 million tons. If you look at the area and productivity of India, Gujarat is the topmost in area, covering 15.35 million hectare, 15.35 lakh hectare. And then if you look at the productivity of groundnut, then Tamil Nadu tops the yield, giving 2691 kg per hectare, and followed by the Rajasthan 2449 kg per hectare. There are quite few constant of groundnut production in India, mostly foliar fungal disease, that is lettuce spot and rust. Then water deficit stress, this occurs due to a lack of proper lack of irregular rainfall, erratic rainfall, and then terminal drought at flowering and harvest stage. Then there are quite few abiotic stresses, biotic and abiotic stresses. So biotic stresses, that is stem rot, collar rot, and peanut butter rust disease. They are also one of the constants in groundnut production in India. Well, oil quality in groundnut, being rich in oil, it has got as many as 13 fatty acids, of which 94% comprises of oleic acid, linoleic acid, palmitic acid, and stearic acid. So if you look at the range of these fatty acids in normal groundnut, so oleic acid contains 36.67%, linoleic acid is 15.43, palmitic acid is 8 to 10%, and stearic is 1 to 4. So here we can see the comparative profile of the groundnut and its counterpart, that is different oils. So if you look at regular groundnut, it has got almost equal amount of polyunsaturated fatty acids and monounsaturated fatty acids. But if we look at other fatty acid, then here we can see polyunsaturated fatty acid, that is linoleic acid, is much more in regular groundnut. Even though it is uh, polyunsaturated fatty acid, it is oxidative instable, due to which it causes the rancidity. And then if you look at the high oleic groundnut, so you can see here, this is the high oleic groundnut, which has got more than 80% oleic acid content. So this is as good as olive oil. We can see here olive oil profile. Here we got about 75% of the oleic acid containing olive oil. So we can see this high oleic groundnut is as good as olive oil in terms of fatty acid profile. If you look at the potential benefits of the high oleic groundnut, it maintains the level of high density lipids in blood, it improves the glucose metabolism, it reduces the triglycerides, it is potential to regulate the appetite, and it suppresses the tumorigenesis. So these are all the benefits of the groundnut with regard to health. If you look at the right side, so there are the industrial benefits of the groundnut, as high oleic groundnut has got about more than 10 folds of the oxidative stability compared to its normal groundnut. Then it is stable, uh, more stable for different products in terms of processing. Then it has got less oil absorption during frying, and it reduces the rancidity during storage. So that's how the high oleic groundnuts are beneficial, not only for the health consumer point of view, but also in terms of industry and then net farm outcome of the farm is for groundnut. So that's how the high oleic is going to be emerging demand of the peanut industry. So oil quality and its importance, if you look at the genetics level, so if you look at genetics level and discovery level, so as such groundnut was almost equal in oleic acid and linoleic acid. But in 1987, there was one discovery where at University of Florida, Gorbett and his co-worker, they found natural mutant. This is phenolic mutant. So it was found that oleic acid content was double than the normal. If you look at here, this phenolic, so this cultivar had about 82% oleic acid than the normal, in which it contains about only 40% of the oleic acid. So that's how the phenolic, that's how the F435-1 was the first groundnut which is natural mutant, and then F435-1 was used for the development of phenolic, and its further groundnut varieties. Here, if you look at the genetics of the high oleic groundnut, so acetyl-CoA is the first stable product of the photosynthesis. So acetyl-CoA gives palmitic acid, and palmitic acid gives the stearic acid. So the conversion of stearic is going to convert into oleic acid by using the different enzymes, series of the enzymes. Here it is the key enzyme which is important for the conversion of oleic into linoleic. Here, if you look at normal peanut, so it has got only 40% peanut, 40% oleic acid, and then uh, as I said, Gorbet they got a natural mutant that is 435-1 and 435-2, in which there was a gene truncation in fatty acid desaturase A and fatty acid desaturase B, in which there was a truncation due to which there was a failure in conversion. It was down regulation, or it was failure in the conversion of oleic to linoleic. So that's how the oleic acid is accumulated more in high oleic peanut, and then linoleic acid was much lesser compared to oleic acid. So here, as I described earlier, it has got two benefits, that is in health benefits and industrial benefit. Health benefits in terms of good fatty acids, then in terms of industrial benefits, highly groundnut got good stability and good oxidative stability, due to which we can store the groundnut for longer period. Not only groundnut but also its derived food products for longer period of time. But that was not the end of oleic acid story. As it happened in other crops, like when we introduce the particular crop for certain industrially or economically important trade, so it has to be very suitable for our ecology. But if we look at this Sunlight 95, when we introduce it into India, it has got spreading types and then long duration, as well as it was not productive in India. And what we want is Spanish bunch type, which are very suitable for Indian conditions. That is, if you can see here, it has got Spanish bunch type, that is bunchy growth, and then it has got high productivity. That's how the challenge was to convert these runner types into the bunch types which are suitable for the Indian ecology. So that's how the breeding for highly groundnut started in ICRISAT back in 2011, where the selection of parents were very key. Looking at the Indian scenario, three parents were involved for the introgression. So ICGV, it stands for ICRISAT groundnut variety. So these were the three parents which were selected for the conversion of low oleic Indian cultivar to conversion of high oleic Indian cultivars. So 6110, it's bold type, then 6142, it was also bold type, and 06420, it was high oil variety. So that's how the initiation of groundnut for high oleic content initiated at ICRISAT back in 2011. Then molecular markers as a very efficient tool of selection was used for the hybridity conformation. As we know that markers, the use of molecular markers in breeding, it is very beneficial for indirect selection of the trait of interest. And then that's how the 64 lines were developed in the background of 6110, 6142, and 06420. Molecular markers were used for the conformation of high oleic content, high oleic alleles in the population. And then seed increase was done during 2013 to 2014 by using NIRS. So I'll explain this in brief, like how this near infrared reflectance spectroscope helps for the fatty acid profile in groundnut. And followed by that, 2014 to 2016 was the multi-location testing at 4 locations for these high oleic peanut lines. And then 2017 to 2018 was the all India coordinated research trialing at 6 locations. And then it was the year 2019, when India got its very first high oleic groundnut, that is Girnar 4, which is ICGV 15083, and Girnar 5, which is ICGV 15093. So both the varieties has got 80% oleic acid content, and these two varieties were identified for cultivation in India. The study was published in Plant Science by Dr. Janila et al. 2016. So this is how the high oleic peanut looks. So this is the Girnar 4 and Girnar 5, and this was the covering by the Hindu, then Times of India, down to Earth. So even though we produce, these two varieties were bred at ICRISAT, that was not the full stop for this high oleic breeding, because because there is huge demand of high oleic peanuts, not only in terms of oil industry but confectionery industry. So what confectionery industry wants is like bigger kernel like this, as well as high oleic content. So for that, there has been attempt to to develop high oleic peanuts along with bold kernel size. So high oleic, it was very important to combine high oleic with resistance to late leaf spot and rust. As in my earlier slide, I explained that late leaf spot and rust. I mentioned late leaf spot and rust are the diseases which occurs in India, and then these two diseases, if come together, they reduces the pod and haulm yield up to 70%. So these two diseases are very devastating, and then if we can see here how it makes impact. This is the susceptible cultivar called Kadri 6, which is a very popular cultivar in India, and this is from the resistant line. So this is how we can see if we bred the variety for resistant to late leaf spot and rust. So this is how it looks like. It will be beneficial for the farmers in terms of pod yield, and then farmers can get the haulm, that is good quality of haulm for their cattle feed. So that's how it is very important to breed the variety, not only for the quality purpose but also for the disease resistance. And as we know that genetic control of the disease resistance is very important, as it is the environment friendly and economically viable method of the controlling disease resistance. So this is a study published in latest publication of our group. It was in Frontiers in Genetics. So here we attempted the improvement of local cultivar called Kadri 6. So Kadri 6, very elite cultivar of the country, India, which covers about 46% of the 46% of the seed demand of the country. Still, it is susceptible for late leaf spot and rust. So we tried to combine late leaf spot and rust and high oleic content in Kadri 6 variety. So what we did is we took Kadri 6 as the recipient parent, and then two varieties. So one is 15033 as a high oleic variety, that is first generation high oleic groundnut variety, which has got 82% oleic acid content, and then we took 13193 as its foliar disease breeding line, which is resistant to late leaf spot and rust. So these two lines we can see here. Kadri 6, it has got very attractive kernel, but it is susceptible for late leaf spot and rust. And this is ground variety which is resistant to late leaf spot and rust, and its variety is 15033. It is a high oleic breeding line. So here it is a farmer showing the produce of Kadri 6. We can see here it is susceptible to late leaf spot and rust. So that's how we targeted this Kadri 6 to improve it for high oleic content and high oleic content and foliar disease resistance, that is late leaf spot and rust. So it started with breeding back in 2016, where we crossed Qadri 6 with 13193, and then we got as many as 210 F1, which were grown in Glasshouse for the generation advancement. Glasshouse provides the window period of good window period, so that we can have 3 generations per year, and we can reduce the breeding cycle per year. So, as I said, like molecular markers, they are very important because we use the DNA of the population to profile it for target trait. It is the indirect way of selection for the presence of allele. It reduces the cost of breeding program, and it is very useful for the confirmation of the target alleles in the breeding population. So here we use 5 markers for the indirect selection by using SNPs, that is single nucleotide polymorphic markers. So here this was the one marker which was responsible for high allelic content, and then these 5 markers, these are the markers which are responsible for laterally spot and rust resistance. So that's how our molecular profiling of for a SNP band. Here, if we look, so homozygous for GG, it means it is susceptible type. It is CC, it is resistant type, and these are the hybrids, that is heterozygotes for the GC. So that's how we use it for confirmation of the target alleles in our breeding population. Apart from that, parallelly, we went for the introduction of high allelic into Qadri 6 variety. So as I said earlier, we used Qadri 6 as a recipient parent and 15033 as a donor parent for high allelic content. And we got 229 F1 seeds, which were again grown in glasshouse, and which was confirmed for hybridity. And then positive plants were tested. And then once we got the F1s from one cross, that is foliar disease resistance cross, that is laterally spot and rust, and once we got the population, that is F1 from high allelic cross, so we intercross these two to get F1s with both the traits. The advantage of having this early generation intercross is we can combine this both the target traits at very early stage of the breeding, so that we can backcross it and we can save the breeding time and resources in the breeding program. So to get that combination, we have to have huge quantity of the F1. So that's how we got 415. We produced 415 F1 plant, and then from which we could get as many as 16 different F2s with different combination. So in summary, this program went like this: Qadri 6 into 15033 and Qadri 6 into 13193. And then we got F1. F1s were intercross to get desirable allele combination in one line or one F1. And then F1s were backcross with Qadri 6 to get BC1 F1, and BC1 F1 were self to get BC1 F2. So that's how the program went. In only a span of 2 years, we could reach up to BC1 F2, and later on, if BC1 F2s were screened for laterally spot and rust resistance by adopting high throughput detached leaf assay, and then evaluation of the oil-qualitative parameter were estimated by using near infrared reflectance spectroscopy, and field evaluation were done for agronomic as well as morphological traits. So, as I said, the field evaluation of the breeding lines were done in the field. At the same time, detached leaf technique, in which we took the media, and then media were with the sand and then vermiculite, and then tissues were quarter fluid tissues were put into the media, and then this tray were put like this as a sample for screening, and then inoculum of laterally spot and rust were sprayed onto the sample, and then this screening or disease assessment was done for little spot and rust in the growth chamber facility at Ikrisand. So if you look at here in our profiling, we got as many as lines for the resident for the little spot and rust. So there are 16 lines. Then out of which this few lines were like as many as 5 lines were resistant to little spot and rust along with other characters. So if you look at Kadri 6 has got 7.5 score. So higher the score, so higher the susceptibility of the genotype for that disease, that is little spot and rust. So here in terms of little spot score, Kadri 6, that is recipient parent, was much higher. That means it was susceptible for the disease. Here, if you see the susceptible cultivar, that is TME2, that was much more susceptible. But if we look at the resistant check, that is GPP4, it has got 3 score, so it was quite good resistance. So we got many lines with almost on par, on equals to GPPT4 in terms of resistance. Later on, second disease was the rust. So if you look at here, we screened these lines for rust resistance, and then we got GPPT4 score with 2.5, and we got many lines with score of about 3 to 4. So the anywhere between 3 to 5 is considered as a resistance. So that's why we got many lines with little spot and rust score. So second objective of the breeding program was to estimate the oil quality of the groundnut. So if you look at oil quality, so it comprises of oil content. So oil content, then 4 different fatty acid, that is oleic acid, linoleic acid, mobile ratio, then palmitic acid, and protein content. So if you look at the biochemical estimation of the groundnut, it takes a lot of time and then resources to estimate the groundnut for quality traits. So what we used in the breeding program is near infrared reflectance spectroscopy. It is a very fantastic tool and an invention of the spectroscopy. That is, it is used near infrared to estimate the quality profile. That too with very very quickly. That is only 80 second it takes to estimate the fatty acid profile and oil quantity, and the two in non-destructive way. I repeat, non-destructive way, because estimation of non-destructive way is very important in the early breeding cycle, as we cannot afford more number of seeds and a time in early testing for the biochemical estimation. That's how the NIRS was very handy to identify the oleic acid in these breeding lines. So if you look at these different lines, so we got many lines with high oleic content. If you look at the higher line, so it was higher check, that is 82.4%. So we got many lines on par or equals to 80%. So as I said earlier, anywhere between 78%, 78% is considered as high oleic content in India. So we got many lines which were more than 78% in this breeding program. Now, we had the target trait tested for little spot and rust. Then we had the testing for oleic acid content and then rust as well. But if you look at the agronomic profile, so these lines were also on par to equals to Kadri 6, that is very popular cultivar. So we had the agronomic profile and then target for the lines which are on par with regard to the different agronomic parameter, that is days to 50% flowering, plant height, then that is number of pods per plant, then that is a CD per plant, and shilling percentage. So we were in search of the lines which are exactly or which looks like Kadri-6 in terms of all the agronomic parameters, and then it is high resistant to foliar diseases, that is lettuce spot and rust, and high oleic content. So here we can see few lines which are resistant to lettuce spot and rust, and then these lines were on par to the Kadri-6 for agromorphological traits. So in terms of farmer preference, it is not only the disease resistance and oil quality, but also the appearance of the pot which matters for the farmers. So there are many criteria wherein farmers will be very precise to select that particular groundnut. So if you look at here, then pod length, pod width, pod constriction, pod reticulation, pod peak, and pod reticulation. These are the quite few parameters which decides the farmer preference for groundnut. And then if you look at here this pod constriction. So pod constriction is not at all preferable for farmer, because when they uproot, so here we can see the constriction, that is gap between two kernels in the same pod. So if you look at here, soil will be adhere here, which creates the problem during processing, because soil will be carrying from with the pod from field to the processing, and then it will be not at all desirable for the farmers. Even here also it is deep constriction. So what farmers need is moderate to slight to the none type of the constriction. And same lies with reticulation. Reticulation is like very strong structure of the on the peanut. So more the reticulation, there is more the lesser the preference. So here what we need is slight to none reticulation. Then here pod big is like this. It looks like weak. So here what happens is if there is a very prominent beak or moderate beak, then beak will get break in the field, which creates the entrance for any fungal pathogen, uh, specifically that is aflatoxin, which is neurotoxin. That's why farmers or the groundnut breeder, they will be looking for the very slight to the absent in the pod beak, and then pod ridges. So there the horizontal lines present, horizontal texture present on the groundnut. So any farmers will be looking for the groundnut with lesser pod ridges, slight pod beak, then moderate constriction and moderate reticulation. So that's how we screen these different lines for pod features, and we got many lines which were slight to moderate in all these pod feature. Now, when we compile together all the traits which we screen, right from the disease score to agronomic trait to pot characteristics to oil quality, we could find three lines which were resistant to lettuce spot and rust, and then which were agronomically preferable, and even in terms of pot characteristics, they were preferable, and most important, they were high in folic acid content. And if you look at the reaction of these superior lines, so here we can see the recipient parent, which is popular cultivar. It has got very susceptible to the lettuce spot and rust. Here it was a donor parent, which was resistant to lettuce spot and rust. And here we can see the lines, they are as good as their donor parents. They are very resistant to lettuce spot and rust disease of the groundnut. And then if you look at the profile of the these lines in terms of folic acid content, so here we can see Kadri-6 as in very first slide, if you can recall. So this red color stands for linoleic acid, and then purple color stands for the palmitic acid. So this both the fatty acids are not preferable for groundnut. So linoleic acid is not preferred because of the oxidative instability of the groundnut and its soil during processing, due to which it creates the rancidity. And here we can see this lesser amount of uh, palmitic and lesser amount of linoleic acid in the groundnut. But if you look at the superior lines, that is over these three lines, so they're as good as their parents, because if you can see here they were high in folic acid content and lesser in linoleic and palmitic acid content. And here, if you look at the protein and oil content, so Kadri-6 has got 49.55% oil content. So these lines were on par to superior with regard to the oil content. If you look at here, so line specifically 278 has got 54.93% oil content. So this can consider as a high oil groundnut in terms of oil content. So this has the combination of high oleic plus high oil content. And if you look at the features of the kernel features and pod feature of the groundnut, so this is the popular cultivar Kadri-6, which we wanted to import for Little Spot and Rust. If you look at the donor parents, they are not much preferable for pod feature and in for kernel feature. But if you look at the lines, if you can see here, they are as good as Kadri-6 in terms of kernels and pod features. Apart from high oleic, so at Ticusat, we are breeding for short duration and high yielding varieties. We also target water deficit stress tolerance groundnut breeding. Then we also target biofortification, that is combination of high iron and zinc plus high oleic variety. And we also target for fresh seed dormancy in Spanish types. And we also target for seed size distribution. Seed size distribution in the sense like in India, there is huge demand for table purpose groundnut, that is bowl type groundnut for confectionery, especially if you look at different products such as salted peanut of many different brands. Then there is very huge demand of the bowl type spinner. So that's how we are looking for the bowl types groundnut. And this is, it has been must have traits in Indian continent because little spot and rust, they are the main constant of the groundnut. So we'll be focusing on the developing varieties resistant to little spot and rust, and we'll also focus on stem rot and collar rot resistant. Among abiotic stress, heat stress, salinity, and then drought is also one of the focus. So if you look at the ground variety developed and released from developed at ICRISAT and released by different partners. So in India, ICRISAT has been the source for direct release of 29 varieties. They are resistant to little spot rust, drought tolerant, qualities such as confectionery, high oleic and high oil, and fresh dormancy. And then in Bangladesh, ICRISAT has provided the base for 6 varieties which are already released. In Myanmar, 7 varieties has been released, and then in Vietnam, 2 varieties were released uh, back in uh, back in late 90s. So it is very important to have the generation advancement in very lesser time. So if you look at different season, so it takes minimum of 7 to 8 years to come up with the variety. So there are few intervention, that is operational practices, which we can use for the operational as operational practices for rapid generation advancement. So here what we did is we try to take a seed chip instead of a leaf chip. So what we use is here seed as a sample for the genotyping. If you can see here, so seed from the posterior end were cut from the posterior end, and it was used as sample for the genotyping. Here we use a seed box, that is a fabric fiber seed box, which was used for keeping the identity as similar as its DNA plate. And then it was used for the profiling of the high oleic content. So here what we tried to do is we tried seed-based genotyping. Here what we get benefits is like we don't need to plant the seeds directly, and here straight away we can reject or select the seeds on the base of its genotypes. So that's how we are having advantage of seed chip for the rapid generation advancement. Apart from this, we got one operational intervention. that is sprouted use of sprouted seeds. What happens generally is we directly go for the sowing. So if we go for the sowing, so sometimes the climate will not be favorable, and sometimes we will get the germination problem as such in the field due to adverse biotic condition. Here what we did is we imbibed the sprouted seeds in warm water, due to which we could get good germination. Even during winter season, we are getting good germination due to uh, due to imbibition of this uh, peanut in warm water. And then these seeds were transplanted directly in the breeding nursery. And then here we can see the nice germination which is coming from the sprouted seed. And then when a groundnut we grow in the glass house, so sometimes we get very less F1s and F2. So to get more number of seeds from the plants which are crossed, it is very important to increase the number of seeds in early generation. For any breeder, it will be very beneficial if we get more number of seeds in F1s and F2s. So here is an attempt to get more number of seeds by using just small intervention, that is use of U-shaped clips. What we did is when we get Spanish bunch type as shown in earlier slides. So Spanish bunch will be like aerial branches. So aerial branches takes a lot of time to emerge the peg from emerge to insert the peg from branch to the soil. So what we did here is we tried to attach the branches to the soil, due to which we could get huge number of F2s from F1s. So these F1s are the reservoir for our future recombinations, and then wherein we can get a nice different combinations, due to which from which we can select good segregants for our target traits. So that's how these three operations were very useful for the rapid generation advancement in ground. I'm thankful to the Palm Learner team for providing me this platform. I'm thankful to Dr. Janila, Principal Scientist, for allowing me to present our fantastic work. I'm thankful to the ground breeding team for the contribution to make the presentation, and then contribution for the development of fantastic ground varieties. I'm thankful to the CRISAT, and I'm thankful to the CRP GLDC Grain Legume Development Center for allowing me to present CRP GLDC, that is the Grain Legume and Talent Cereal, for providing me the platform to present this research. And then I'll be happy to take any question if there is any. I'm reachable uh, to the my email ID, which is mentioned here. Thank you so much. So you can reward me back with this email ID.
and then finally for genome association study for disease resistant traits in barley. So I will explain in next slide how the data, how this genotyping sequencing works, and how that data can be used for doing all this downstream analysis. So genotyping by sequencing, as the name suggests, it's the procedure for simultaneous SNP identification and genotyping of large number of samples from population. So in this genotyping by sequencing, first step is to extract the high quality DNA from the given species, and then the DNA is restriction digested with different restriction enzymes. And then this restriction digestion can be carried out either using single enzyme or with double enzyme. It depends based on the complexity of your species. Such restriction digested products are then ligated with the two forward and reverse adapters. The forward adapter has one unique DNA barcode of 6 base pair length, and this is unique for each sample. So in this way, you can combine, you can extract DNA from different samples or digest using the restriction enzyme and attach barcodes which will be unique for each sample. And in this way, you can such restriction digested samples can be pooled and then sequenced. After sequencing, you can separate the reads based on this unique DNA barcode. And then, yeah, the reads can be used for identification of SNPs, and then the SNP data can be used for further downstream studies. So yeah, based on the different technology, the read length can vary. For Illumina, it's just generally 250 base pair. So yeah, these are very short reads. And then first step in this read processing is to remove the barcodes or the adapters which we added during restriction digestion process. And also there will be some bases which are of low quality, and then you have to remove such bases also. So there are various softwares available for doing this. And then this cutout is the one of the widely used software for adapter and quality trimming. And then the quality of reads can be visualized using different softwares. And the FastQC is one of such software for visualizing the read quality. And the next step in this SNP identification is to map these high quality reads against the reference genome sequence. Yeah, and then again, there are different softwares available, such as BWA and then NovaSort. And then, yeah, you can again check the mapping quality using different softwares. And only those reads which are mapped at high quality are used for SNP identification. Here we use SAMTools, but yeah, there are other softwares such as GATK, which can also be used for SNP identification. And then you can output the SNP matrix in various formats such as VCF, HapMap, or Blink. These are the widely used format for the storing the SNP data. Here, I'm just showing one of the output of the SNP identification pipeline. So you can see these are this is the genome of my species. It has basically 21 chromosomes, and these are labeled as 1A, 1B, 1T, 2A, 2B, 2D, and then this is the length of each chromosome in MB, and you can see how many SNPs you get from each chromosome. So at 10% missing data, I got around 23,000 SNPs, while at 20% missing data, I got around 40,000 SNPs. And again, here it shows the distribution of SNPs on the genome. And as you can see, there are more SNPs towards the end of the chromosomes. This is because we use a methylation sensitive restriction enzymes, and the center part is highly methylated. And so the restriction enzymes, they cannot able to cut in the center part. While also the there is very high recombination rate towards the end. And so the SNP density is also high towards the end. Yeah, and the first use of this SNP data is to study the genetic diversity. There are different methods available for studying the genetic diversity. One of the widely used method is principal component analysis. And here you can see the principal component analysis plot for around 6,000 samples. Each dot is one sample, and then I have colored them based on the different geographic origin. And you can see the samples from European, they have clustered together. Similarly, sample from Asian region, they have also clustered together. So it means these samples from Asia, they are closely related among themselves, but they are distantly related to the samples from European region. Yeah, so this way you can, when you have some samples with unknown origin, you can check, you can predict from which region it might have came. Second important things for this the SNP data is you can identify the duplicate sample. So this is just like you are comparing SNP marker from one sample against another, and then check how many SNPs are common between these two markers, and based on that you can predict or determine how closely related these samples are. And then one of the method used for identifying the duplicates is calculation of this IBS. It's the identity by state matrix, which is nothing but it shows how these SNPs are related to each other. And higher the value means higher the samples are related to each other. So if you say this 85 means, okay, 85% genome of these two samples are similar, while 1 means these two samples are highly similar or identical. And then you can, based on this plot, you can identify such duplicate samples, and then you can decide what to do. You can either remove them from the downstream analysis, or Another important aspect of this genotyping or SNP analysis is doing genome association study. So this is basically studying how your genotypic data is related to the phenotypic data. And then for this, you have to first sequence diverse collection, like we sequence around 3,000 samples from one species. And then we got 20,000 SNPs with 10% missing data. And then we also phenotype these 3,000 samples for one of the important diseases. And then you combine this genotypic and phenotypic data, and then run the genome association analysis. There are different models available for doing this, like generalized linear model, mixed linear model, and so on and so forth. So we use this mixed linear model, which also correct for the population structure. And then we did the GWAS using GAPIT software. And yeah, the result of GWAS can be visualized using this Manhattan plot. And yeah, you can see these are again the 21 chromosomes from my species. And yeah, from this plot you can see there is no significant SNPs for this disease, because this is mainly because disease resistance is very complex trait, and it's not possible to identify candidate loci with only 20,000 SNP markers. And so we need more number of markers, and yeah, GBS might not be useful for identifying the candidate loci for this disease resistant genes. So people are nowadays people are using another methods. It's called association genetics with resistant gene enrichment sequencing. So yeah, in plant there are different kind of resistant genes present. They act against the pathogen, and then yeah, somehow prevent plant from getting infected. So majority of cloned R genes are of NBS LRR class. There are different types of resistant gene present. but the majority of genes are operated in base LRR class. And then, yeah, there were different approaches used for identifying of resistant genes, such as map-based cloning, where you have to develop a large population, do QTL analysis, and then find map this population, and then identify the candidate genes. And another method is transposon tagging, but these two methods are very time consuming, and yeah, it requires at least 5-6 years of hard work, and they are also laborious. So recently, the Jupe et al. They developed a resistant gene enrichment sequencing method. So this method uses this 120 base pair RNA sequences. These are called BETs. So these BETs, they bind to these are complementary to the NBSLR sequences, and these are biorelated towards the 3' end, and then this can be captured using tryptophan-coated magnetic beads. But yeah, though this resistant gene enrichment sequencing is very useful, we cannot use for doing we cannot use this for sequencing 3,000 samples, and so we have to you have to identify some diverse accessions. So again, this is the use of this principal component analysis. Yeah, from the principal component analysis, you can select diverse accessions, and which are covering the entire diversity also. Yeah, and here we selected those samples with this yellow and pink color for these different diseases. And so from this 3,000, we selected 800 samples, which are really diverse and correct, and also capture the entire diversity. So this resistant gene enrichment sequencing, it is divided into two main parts. First is library preparation, and then another is the hybridization. Again, and this library preparation part is further divided into three parts. First, you have to extract the DNA and then fragment the DNA. And then this fragmented DNA is ligated with the same way like we did it in the genotyping by sequencing. So these are again ligated with the adapters, and then each adapter, one forward adapter has one specific barcode sequence. And then you amplify this product using index PCR. Once you amplify the products, you can combine the samples from DNA from different samples, and this is called library multiplexing. And then from this multiplex library, you can capture the DNA fragments using the biotinted probes, and then this can be captured using tryptophan-coated magnetic beads. Yeah, and then so this is the important step. Here you are only capturing fragments which are from this NBSLRR gene, and then you are again amplifying these sample fragments, and then you can sequence based on the different platforms such as PacBio or Illumina. Yeah, so this is once you do the sequencing, you will get the short reads because we use this Illumina. So maximum read length was 250 base pair, and so we did pair end sequencing. So so maximum the fragment what we can sequence is of 500 base pair length. Yeah, again you have to remove the adapters and do the quality trimming. Once these reads are trimmed, you can, instead of calling for SNPs, here you identify KMER. So KMER are nothing but short stretch of sequences, and these are, and then this can act as also the markers. So you identify these KMERs from all the reads from each sample, and then you will get this KMER count table. So this is nothing but the different KMERs and how many times this particular KMER has occurred in your sample. And then you repeat this KMER counting process for all the samples, and then you check, okay, in which sample this particular KMER is present. So so for example, for this S1 sample, the KMER 1, 3, 4, and N is present, but this KMER 2 is absent in S1 sample. Similarly, for S3 sample, KMER1 is absent, but other K2, 3, 4, and N KMERs are present. So this is the same matrix like SNPs. So this is called present substance matrix. And this matrix, along with phenotyping data, can be used for doing the genome association studies. And then, yeah, again, you identify KMERs which are associated with the trait of interest. And these KMERs, they can be mapped on the reference genome, or you can also develop a de novo assembly based on the reads, based on this NBS RenSeq reads for one of the registration samples. And then these KMERs can also be mapped against the quantic assembly of the resistance samples. And then based on that, you identify quantics from which maximum number of KMERs are associated with a particular trait of interest. And then this candidate may be the probable complete NBS-LRR gene responsible for that disease. So we did this registration gene enrichment sequencing for one of the diseases in our PRECISE. And here you can see this is the very, saturated graph. But on the right side, you can see there were 3 quantics from which a large number of KMERs were significant. And these 3 quantics, out of these 3 quantics, 1 quantic was having the full length coiled NBS-LRR gene, while 2 were the partial quantics. So yeah, you can see from this, we can rapidly identify the full length NBS-LRR gene responsible for the disease resistance. So in summary, yeah, genomics, especially NGS technology, can be used for faster and efficient fingerprinting of large number of samples, and then also for identification of duplicates and for removing the redundancy, then selection of diverse samples like we did. Out from these 3,000, we selected 800 diverse samples, and also for genome association study and for genomic selection analysis. And then I also showed you that registration gene enrichment sequencing, how registration gene enrichment sequencing can be used for rapid identification candidate genes. And then these candidate genes can be used for improving the disease resistance rate from that particular crop. Yeah, as you can see, this is so much of work, and yeah, I would like to thanks my all the colleagues and my supervisors from IPK Garters 11. And finally, thank you very much for your listening. Bye-bye.